Hey guys, you're back, and I'm back because I'm supercharged about this. Woo, I was procrastinating and we're getting it done, and it's like, hey, this actually isn't so bad. Um, Other than all the Twizzlers stuck in my teeth. Yes, I totally sucked down a bunch more. My little girl is still sleeping on her bed. It's a beautiful sunny Sunday, and I am recording lectures. But we're almost through with chapter 12. This is lecture 3. This might be the last one. I need to keep a better eye on the clock. I should have made that one, the last one, a little bit shorter. Sorry about that, but once you get started, I just can't stop. It was technical. It was difficult. Hopefully you're looking at some of that other resource material I put on Wiley Plus to help with some of these concepts. But it's really just all about sodium and potassium and being at threshold. And a bunch of vocabulary. So I think you guys can handle it. I'm feeling pretty good. I hope you are too. I think about all the people that I don't see. I love seeing you guys in lab. If you watch these, I hope you guys are doing okay. The ones that aren't able to see me in person at all. All right. So when we last talked about in lecture 2 on this chapter, with the sodium rushing in and the potassium leaving, and too much potassium leaving, the hyperpolarizing, we were talking about the fact that action potentials can only go one direction, and they can only go one direction because once the sodium gates open, it kind of closes weird, and it takes a little bit for them to get fixed. So it can't go back because they're still like stalled out. So it will only go one direction, down an axon. And what happens is, if this is my neuron, it has to go the full length of every sodium potassium channel that's along that whole axon. So if it starts here, then it goes there, and it'll move along the whole length. If I have to go the whole length, that is known as continuous conduction. I have to stop. It's like when you take your dog for a walk and your dog stops every 2 feet to smell a pole, a piece of grass, whatever. It takes forever to walk down the street. Now, if you're like, okay, dog, you're only checking out the telephone poles that are spaced 500 yards apiece. Oops. You're jumping from gap to gap. And that's kind of what happens in neurons. If I have myelin on my axons, if my axons are myelinated, and then I have these spaces called nodes of Ranvier, this action potential will jump from gap to gap to gap. All right, so it's saying that here it's here it's there it's there. So instead of going the whole length, there's another, I just go gap to gap to gap to gap. And that is called saltatory conduction. So this is awesome in all things and happy unless you have MS. When you have MS and your myelin sheaths break down, then your conduction, sodium potassium channels grow where there is no myelin, and you will go from saltatory conduction to continuous conduction if you have multiple sclerosis and your myelin sheaths are munched away. Ugh. Whatever, degraded. Oh my goodness, computer. I'm about to scream. Myelin sheath degraded. So saltatory is what you want. And, so here we go. Types of nerve impulse like routes. You can have the local or you can have the expressway. I can have continuous, occurs only in unmyelinated, and it's step-by-step depolarization and repolarization of every part of the axon. Saltatory conduction is only if I'm myelinated, much faster, of course, because it's only at jumping from node to node to node of the nodes of Ranvier, which are the gaps in the myelin sheath. So it's less overall movement of sodium potassium, less ATP is being used, and it's quicker. So that is going to be one factor that affects my speed of my impulse, my propagation speed. One is my amount of myelin. The other is temperature. Hotter is faster. And the other is the axon diameter. Larger diameter are quicker. So when we put it all together, well, factors that affect the speed. Oh, we just talked about that. I have 3 different types of fibers. I have a group A, I have a group B, and I have a group C. So group A is my fastest. They're heavily myelinated, large diameter, heavily myelinated. Somatic sensory, so this is my input. And my motor fibers, my output of my somatic system, right? My voluntary system, and some pain and some temperature. They transmit at 130 meters a second. What is that? How many feet is 130 meters? I'm about to look it up because I can't do the math. I guess I'm gonna have to Google it because I can't. I'm blanked out. How many feet in 130 meters? 426 feet. 426 feet a second is how fast impulses travel on those fibers. And then it hits axon terminal, and then I'm going to depend on diffusion of neurotransmitter to then send it again. That makes me laugh. Group B are intermediate. Where's my little writing tool? Intermediate diameter, some myelin, lightly myelinated. And this is my sensory inside the body, visceral sensory, so that's inside body. And all my autonomic motor neurons, and these are talking to smooth muscle, cardiac muscle, and glands. So those guys aren't as quick as the output talking to skeletal muscles, because those are group A. And group C are the smallest, so they transmit at 15 meters per second. Group C, the smallest in diameter, unmyelinated. They're unmyelinated, and that's typically all of the just stuff inside the body, autonomic nervous system. Now it's some sensory and pain, and these transmit at, look at that, 2 meters per second, right? What? So how many meters? I don't remember. A lot slower than 130 meters, so that's for sure. And what this is like if you cut yourself, you feel pain instantaneously. That's stimulating my group A fibers. But later on, you still have that itching, burning pain. That's your group C fibers. Kind of crazy why you feel different pain during it, right? Because, we have cats and they scratch us if we get too obnoxious with them. And, right, you first get it and you're like, ah! And then, it's throbbing and it's itching and it's weird later on. I just thought that was kind of interesting. So, different factors affect the rate of propagation of the impulse. The axon diameter, temperature, we'll never talk about, and the amount of myelination. So I have my group A, my group B, and my group C. Encoding of intensity, we already talked about that. This is old stuff. But this is a cool graphic, and this is actually from a different book. This one's not in your book. So first I'm looking at this. This is my stimulus voltage, because I'll just tap something. And if it's below threshold, I don't get any response. If my stimulus is above threshold, I'll get my action potential. As my stimulus strength increases, it has increased my frequency of my action potential. So increased strength will increase frequency of, oh my gosh, you guys must hate my handwriting. I really don't handwrite that bad. You'd be surprised. Okay. What time is it? It's only 10 minutes, so I'm going to keep talking. So once I have all this going on with the neuron, well, what do neurons do? A neuron's either going to talk to another neuron or talk to an effector, and it can talk to them one of two ways. It can actually be in contact, connected via gap junction. That's an electrical synapse. That's not that common. The majority of synapses we have, there is the synaptic cleft space we saw, and that's a chemical synapse. So I have a synaptic cleft, and I will have neurotransmitter. I am sucking whatchamacallit out of my teeth, by the way. If you see me looking weird, it's because I got a little Twizzler going on. Okay, so it's one way from, so when I have two neurons, this is going to actually come into play much later, right? I had this space, that's the synapse. That's the space between. I will have the presynaptic, the guy before neuron. I will have, and that's that guy, because the impulse is going that direction, and I will have the postsynaptic. You know what I'm trying to say. All right, so I have the presynaptic, guy number 1, postsynaptic, guy number 2, right? And I have the synapse between the two of them. So synapses can go, this is just a waste of time. I can be an axon talking to another axon, so it's exoaxonic. I can be an axon going to a dendrite, that's exodendritic, or an axon going to the cell body or soma, so that's axosomatic. That doesn't even really matter. But what comes back into play is this chemical synapse. So we recognize this, hopefully. This is saying that it's a presynaptic neuron. This is the synaptic terminal, the end bulb, full of synaptic vesicles with neurotransmitter, right? We saw this already in the neuromuscular junction with acetylcholine. So as the impulse is shooting down, it is causing calcium channels to open on the presynaptic neuron. Calcium is going into the cell and it's pushing out these vesicles full of neurotransmitter, exocytosis. They're leaving the cell. They dump neurotransmitter into this synaptic cleft. That's the space. Neurotransmitter will diffuse across the space. Diffuses across the cleft, and then it will bind. All right, so the neurotransmitter will bind with receptors on the postsynaptic neuron. We had it binding with receptors on the sarcolemma, on the muscle cell, but it's all the same. So when it binds, it opens up that gate and then sodium will enter in. So that's a ligand-gated channel because it had to bind with the neurotransmitter. It then opens it up. Sodium rushes in to the guy number 2, postsynaptic neuron, and then I will have another action potential. Action potential is continued. Generated and continued. Generated on guy number 2. So we've already seen this. This is not new information. If it is, I'm sorry. I'm not going to go over it again because this will be like the 3rd time I've talked about a chemical synapse. But let's see. These are all the words. Let's see. Nerve impulse arrives at synaptic end bulb. When it's getting there, the calcium gates are opening, which is triggering exocytosis. Let's see, what was guy number 3? Triggering exocytosis of the vesicles filled with neurotransmitter. They're released into the cleft, diffuse across. The cleft diffuses everywhere, right? Diffusion's random. Will bind with receptors on the postsynaptic neuron, opening up those channels. Sodium will rush in to the postsynaptic, into the post-guy, and will create the action potential if it's at an appropriate level. Whoops, I just wanted to go. Oh, I guess that was 6 and 7. So those were the numbers that went with this little picture, if I talked too fast. All right. So guy number 2, now this is just an interesting concept. We're not going to spend a lot of time on it, right? This guy number 2, postsynaptic neuron. Well, let's just call him guy number 2. Guy number 1 was the pre, right? Well, guy number 1, when he's sending guy number 2 the message, he can either turn him on and it be excitatory, or he can turn him off and it be inhibitory. That's a weird concept. I don't want to spend a lot of time on that. We're not going to go into that, but that's a lot about how vision works. Vision is a 3-neuron chain, and some get turned on, some get turned off, and then that's only just in your retina, not even talking about what's going on in the cortex in your brain. So if he's being excitatory, he is going to depolarize. This aggravates me so much. Or he can make the guy more negative, hyperpolarize, and that is inhibitory. Again, I don't want to go into that, but that's more of like that, what is it? Neuropsychology and that sort of thing. So once neurotransmitter is done, I need to get rid of it. Well, we've seen one way of it being gone. Otherwise, it will continue to send a message. So it needs to be removed, and I can remove it by an enzyme like acetylcholinesterase. Removed degraded acetylcholine. I can actively suck it back up, reuptake, and then recycle it. Right? That's kind of like, I can't even use the word. It's inappropriate at this point. When you give somebody something and then you take it back. You're like, oh, I didn't mean that. Oh, give that back to me. And that's reuptake, or it just kind of diffuses away into no man's land. Okay. Alrighty. So I'm thinking that a lot. I hate to lose them all, but bye. Where are we? A bunch of these slides are about summation, temporal and spatial. Now, we're not talking about that. I'm never going to ask you a question about that. I left in here for you guys all this content on neurotransmitter because there's all these different types of neurotransmitter. We're not going to be talking about it in lecture. I'm not going to test you on it, but I left it for you for reference, especially you psychology majors. Right, so there is neurotransmitter acetylcholine, amino acids, biogenic amines, ATP and other purines, nitric acid, and even carbon monoxide. So it kind of talks about different types. You guys would know about them. Neural circuits we're not going to talk about as well. But I might as well. What time am I up to here? Oh, I wanted to talk about some of this. Oh yeah, plenty of time. Just finish this with lecture number 3. Put this chapter to bed. Regeneration and repair of nervous tissue. their neurons have a very limited ability to regenerate. But they can change their shape. I've never even talked about this plasticity. The capability to change based on the experience. Sprouting new dendrites, new proteins, changed contacts. Okay. But I'm not able to replace myself. In the central nervous system, oh, this is what I was talking about. There's little to no repair in the brain and spinal cord. But because of the axons are unable to fix. And look, this is the deal. The oligodendrocytes inhibit regrowth. So it was in the central nervous system, not the peripheral. My bad. They're like, the big brother bully. And astrocytes will, near the damage, will form scar tissue barrier. So scar tissue is non-functional. So you kind of have the odds stacked against you between your astrocytes and your oligodendrocytes will just not allow for a repair. This is cool. New neurons are able to arise in the hippocampus. I won't talk about the brain that much because I don't like it. I don't have other growth stimulating cues, and scar tissue is a lot easier to plug into the holes, which is non-functional. In the peripheral nervous system, what does this say? I've typed this up because I looked it up a different time, but I'm not totally prepared to talk about this. So repair is possible if the cell body is intact, and if the Schwann cells are functional, and scar tissue doesn't form too rapidly. That's a lot of ducks to have in a row in order for repair to be possible. I'm going to have, oh my gosh, all of this crazy specific big word action. Chromatolysis in the cell body, with the Nissl bodies, the rough endoplasmic reticulum break up into masses. Oh my gosh. I don't even want to talk about this one. It's too crazy. Damage and repair. Oh, hello. So here's a normal neuron. That's what I like looking at. So in order for repair to happen, it looks like I gotta have some good Schwann cells, some good Wallerian degeneration, that's an organized degeneration, to then just repair it all up. I don't know. I'm not comfortable thinking about that. But there were a lot of disorders, and your book, your e-text actually talks about a lot, and I thought that were pretty interesting. There's multiple sclerosis. A lot of people did papers on epilepsy, although heck of a lot did Addison's disease. Depression, neuroblastoma, rabies, and Guillain-Barré syndrome are also mentioned in your textbook. I threw in a few slides with multiple sclerosis, where it's an autoimmune disorder where your myelin sheath is munched up, and therefore you have cross circuitry, like, signals cross circuits, and it just becomes all jumbled and disorganized because of the lack of that insulation, that protective insulation barrier of the myelin sheath. Some people did it on epilepsy. There's a localized focal point of just circuitry going everywhere versus generalized throughout your brain. Look at, here's your normal brain activity, but if it's epileptic, you just have firings and nerve impulses just everywhere scattered, hence, just a seizure. Oh, all right. So that is the end of chapter 12. A little technical, a little not technical, some putting pieces together, some miscellaneous bits, but you guys got it. So I'm always curious at who's watching these things. I never know. Anyway, that's all I have. So email me if you have questions. Work on, oh, you know what? Let's see. I'm going to go tiny again because I want to look at the chapter objectives for chapter 12. I have a few minutes. Okay. Did we cover these learning objectives? 3 basic functions: sensory, integrative, motor, organization, structural and or functional. Describe a neuron, be able to label it. What is it for? Oh, that's the structurally and functionally. Oh, the organization of the nervous system, that's the central and the peripheral. Peripheral breaking up. Okay, the neuroglia and their functions, importance of myelin, the cellular properties that permit communication. I don't even know what to do with that one. That one should come out. That's weird. The types of ion channels. How do I create a resting membrane potential? Right? That's where's sodium, where's potassium, why is sodium where it is, why is potassium where it is, who's maintaining it, the parts of that action potential, how it's propagated along neurons. Really, I'm looking for that one. I don't know. Absolute and relative refractory period, saltatory, continuous conduction, factors that affect the speed, class A, B, and C. A synapse. Talk about a chemical synapse and what's happening versus an electrical is just a gap junction. And then how do I remove neurotransmitter from the area? So focus on those questions. There's a few I would delete if I had, I don't want to delete it now because then you'll go back to us. You're like, well, they're not even there. Anyway, that I think is it. So I'm having you do all this stuff, all these little interactivities, reinforce some of the concepts, and then you have a few little assignments to work the material. All right, my friends. I guess I'm going to call a ball. I kind of miss you. I will see you for talking about the brain. That'll be next weekend. So take care. Take care of yourself. And I'll see you. See you on the flip side. Bye.